Welcome back to Unit 2. Today we're going to continue on topics 1 to 10 and 11, or cell structure and function. Last time we were talking about the endomembrane organelles. Today we're going to talk about the energy organelles as well as components of the cytoskeleton. So let's go ahead and get started with the endosymbiont theory first. So the endosymbiont theory is the theory that explains the similarities mitochondria and chloroplasts have to a prokaryote. So this theory states that an early eukaryotic cell engulfed a prokaryotic cell. And then what happened is the prokaryotic cell became an endosymbiont, or a cell that lives in another cell. So basically, the theory is saying that when the eukaryotic cell engulfed the prokaryotic cell, it was a mutualistic relationship. So they benefited each other by being one. And so therefore, they became one functional organism. Now, if you're thinking, how did they come up with this or what evidence supports it? There's a few pieces of evidence that point to this theory or that support this theory. So firstly, mitochondria and chloroplasts, they both have a double membrane. Mitochondria and chloroplasts also have their own ribosomes. They have circular DNA, which, if you know prokaryotes, right? Prokaryotes have circular DNA, and they're capable of functioning on their own. Now, even though they're capable of functioning on their own, we actually don't see, free mitochondria or free chloroplasts running about on their own. They're only found now in eukaryotes. However, they would be, since they have their own circular DNA, their own ribosomes, right? They're double membrane. They would technically be capable of functioning on their own. So those pieces of evidence support the endosymbiont theory. Now let's go ahead and go through the mitochondria and chloroplasts and talk more about their specific functions. Let's start with the mitochondria. So the mitochondria is going to be the site of cellular respiration. So it has a double membrane. So what's the structure of the double membrane? Well, the outer membrane is smooth, but the inner membrane has folds called cristae. And this is what allows for compartmentalization in the mitochondria. So the cristae is going to divide the mitochondria into two internal compartments. And remember, compartmentalization increases surface area. So if the cristae is a form of compartmentalization, we'll see an increase in surface area. Let's go through the intermembrane and the mitochondrial matrix now. So the intermembrane is the space between the inner and outer membrane. And you can see that there, that light orange color. The mitochondrial matrix is enclosed by the inner membrane. This is a location for the Krebs cycle, and it's going to contain a few different important things. So it's firstly going to contain enzymes that catalyze cellular respiration and produce ATP, which is our energy. It'll contain the mitochondrial DNA, and then it'll also contain ribosomes. Now, cells can have a different number of mitochondria, and depending upon their metabolic activity, or their metabolic activity determines how many mitochondria they have. So a cell with high metabolic activity would have more mitochondria. So for example, cells that contract or cells that move, think muscle cells. Muscle cells contract. Since they're going through that movement, they're going to have, muscle cells, more mitochondria than other cells in the body. All right, that's mitochondria though. Now let's look at the chloroplasts. So chloroplasts are specialized organelles in photosynthetic organisms, so think plants. They're the site of photosynthesis, and they're green because they contain the green pigment chlorophyll. And we'll talk about the importance of chlorophyll and pigments a little bit more in Unit 3. So what's inside of its double membrane? Well, firstly, we're going to have thylakoids, and we can see thylakoids are those thickened parts there in that first image. These are membranous sacs that can organize into stacks called grana. And we can see the grana right there. And the grana are important because they're going to be the location of the light-dependent reactions. And that's an important component of photosynthesis. Now, also in the chloroplast, we have the stroma, which is like the cytoplasm for the chloroplast. So this is a fluid around the thylakoids. And this is important because it's a location for the Calvin cycle. It's also going to contain the chloroplast DNA, the chloroplast ribosomes, and also important enzymes for photosynthesis. All right, that's it though for the energy organelles. So now we've covered all of our organelles, and now we're going to delve into components of the cytoskeleton. So to start off, the cytoskeleton is just a network of fibers throughout the cytoplasm that give both structural and mechanical support, especially for animal cells. Just some general functions. They're going to be used for anchoring of organelles to make sure that organelles stay in a relatively the same location. They can allow for movement of vesicles and also some organelles and even the whole cell. And movement, just in general, occurs when the cytoskeleton interacts with motor proteins. We're going to discuss the 3 types of fibers that you can find in the cytoskeleton next. So the 3 fibers that you can find are microtubules, microfilaments, and then intermediate filaments. And we're going to go through these each, starting with microtubules. So microtubules, these are hollow, rod-like structures made from the protein tubulin. These grow from the centrosome, which we saw in the last video at the animal cell. That's a unique component for our animal cell. So they're going to grow from the centrosome, and they're going to assist in microtubule assembly. All right, so that's general. Now let's look at some specific functions. So firstly, they can serve as structural support, and I like to think of them as almost tracks for movement of organelles that are interacting with motor proteins. So if an organelle comes into contact or interacts with a motor protein, microtubules are like the tracks for the train, basically, and they'll move on the microtubules. They can also assist in the separation of chromosomes during cell division and also cell motility in think cilia and flagella, right? Those are the two options for cells for movement. All right, next we have microfilaments. Microfilaments are thin, solid rods made of the protein actin. Actin is especially important in muscle cells, and we'll talk about that in just a second. What are the functions of microfilaments? Well, firstly, they will help maintain cell shape by bearing tension. They can assist in muscle contraction, again, because they have actin, and cell motility. And specifically how they do this is actin works with another protein called myosin, and together actin and myosin cause a contraction in your muscle cells. And then lastly, the division of animal cells by helping to form the contractile ring of the cleavage furrow, which we'll look at when we get into cell division. And lastly, we have intermediate filaments. Intermediate filaments are fibrous proteins made up of varying subunits, and these are permanent structural elements of cells, and they have a few important functions. Firstly, they help to maintain cell shape. They anchor the nucleus as well as other organelles, and then they're going to form the nuclear lamina, which is what lines the nuclear envelope. All right, but that's it for the cytoskeleton. You have a practice FRQ here, so go ahead, read through this, and I want you to formulate a response in your workbook, and then we'll go over that together in class.